All right, hello everyone. My name is Jackson. I'm a new tutor here at Premed Peers. We're going to go through a bio passage together. All right, so let's get this figured out. All right, this passage is from Jack Weston. I'll read it through with you guys and we'll go through it slowly. All right, so despite their hard, persistent nature and relatively static proportions, human bones are an extraordinarily dynamic tissue that is continuously resorbed, remodeled, and renewed. The maintenance of these processes requires precisely regulated homeostatic mechanisms in order to preserve adequate bone strength, structure, and density. Bone resorption is triggered by parathyroid hormone. Under conditions of low calcium, parathyroid hormone will bind to receptors on osteoblasts and upregulate the expression of the signaling protein RANKL. RANKL binds to the receptor RANK on osteoclast precursors, activating the transcription factor NF-KB, which signals them to differentiate and activate into functioning osteoclasts. OPG, produced in osteoblasts, competitively blocks RANKL, thereby preventing signaling. Because of this, net bone formation or resorption can be said to be a function of the ratio of OPG to RANKL. Osteoblasts synthesize and arrange bone matrix proteins, their chief role being to deposit collagen and other proteins to form new bone. These important interactions are shown in Figure 1. Several diseases are known in which some part of this process has been disturbed and thus proceeds out of balance. Osteoporosis is a disease characterized by decreased bone density and a change in bone structure wherein the bones become increasingly porous. Osteoporosis affects over half the population of the United States over 50 years old and is overwhelmingly more common among postmenopausal women, largely due to the marked decrease in estrogen levels. Estrogen plays an important role in regulating levels of RANKL and OPG. Autosomal recessive osteopetrosis, or ARO, on the other hand, often referred to as marble bone disease, is characterized by excessively thick and dense, albeit brittle bones. ARO is a congenital condition affecting individuals from birth due to a number of genetic anomalies, typically resulting in death by the age of 10. One variant results from a mutation in the gene coding for RANKL, others from defects in OPG. A hypothetical study hopes to determine the epidemiology of osteoporosis and RAO in the United States and attaches a survey to annual political election ballots routinely over 10 years, with a yearly sample size of nearly 10 million. The survey asks simple yes/no questions regarding if the individual, the voter, has ever had either of the two conditions. The results are shown in Table 1. And here we have Table 1. All right. So the first thing that we need to do is we need to pick out a little bit of strategy when reading this passage. I personally like to skim the passage, then read the first question, and I go back and pick out the necessary parts of the question that I need to answer the question, so I'm not overwhelmed by the entirety of the information within the passage. However, you might have a different strategy, so I invite you to use what works best for you. But nonetheless, we're going to get started with the first question. So question 1 says, assuming each disease is due to a single defect in cells involved in bone turnover, what are the most likely defects in osteoporosis and osteopetrosis, respectively? Okay, so here we're looking at osteoporosis and osteopetrosis diseases as a result of a defect in a single cell. Okay, so what we need to look at first is the parts of the question that have to do with osteoporosis and osteopetrosis, or the passage, excuse me. So osteoporosis here is a disease characterized by decreased bone density and a change in bone structure wherein the bone becomes increasingly porous. So osteoporosis leads to brittle bones or increased bone resorption, whereas osteopetrosis, which it says here, is characterized by excessively thick and dense, albeit brittle bones. So these are essentially the opposite presenting diseases. So we need to look at an answer that's going to give us essentially the opposite result. So if we look at here, A, osteoporosis is due to the increased function of osteoblasts, whereas osteopetrosis is due to the decreased function of osteoblasts. So the key here is increase versus decrease. We're looking at the same type of cell, osteoblasts, instead of looking at both osteoblasts and osteoclasts, which are two different types of cells. If you look at B here, osteoporosis is due to the increased function of osteoclasts, and osteopetrosis is due to the decreased function of osteoclasts. So these two answers, A and B, are essentially saying the same thing with osteoblasts and osteoclasts. So we need to determine the function of both osteoblasts and osteoclasts. So we can see from this this diagram here, osteoblasts lead to bone formation, whereas osteoclasts lead to bone resorption. A good mnemonic for this is osteoblasts build bone and osteoclasts resorb bone, or chew bone, osteoclasts chew bone. So if we think of this in terms of the question, osteoporosis is due to the increased function of osteoclasts. So increasing osteoclasts will cause an increase in bone resorption, whereas osteopetrosis, if it's due to the decreased function of osteoclasts, osteopetrosis will result in less bone resorption. Whereas A is saying the exact opposite, osteoporosis is due to the increased function of osteoblasts, or increased bone formation, and osteopetrosis is due to the decreased function of osteoblasts, or decreased bone formation. So as we said earlier, osteoporosis results in brittle bones, or excuse me, not brittle bones, but porous bones. So essentially, we're looking at bone resorption. So that means that we know right away that A cannot be right, because osteoporosis, which is marked by increased bone resorption, would not have increased function of osteoblasts, because that would cause increased bone formation. So we can already rule out A. C, on the other hand, says both osteoporosis and osteopetrosis are due to the decreased function of osteoclasts. We can instantly knock this one out, just because we know that osteoclasts, or osteoporosis and osteopetrosis, are essentially the opposite disease, so they cannot be caused by the same decrease in function of osteoclasts, of this one type of bone resorption cell. And then if we look at D, it's essentially saying the exact same thing. Here we're saying increased function of osteoblasts, excuse me, which would have the net result as C, of decreased function of osteoclasts, because osteoclasts and osteoblasts are the exact opposite of each other. So we know C and D are both not correct. We already ruled out A, so let's look at B. Osteoporosis is due to the increased function of osteoclasts, whereas osteopetrosis is due to the decreased function of osteoclasts. So osteoporosis, or porous bone, could result from increased function of osteoclasts, because we know osteoclasts resorb bone. Osteopetrosis is the opposite. They're dense, hard, brittle bone. That also could happen from decreased function of osteoclasts. So we know that B must be our answer here. And here we know that it is our answer. If I go over here, I've written out, I've written out the mnemonic that I use to help me determine the function of osteoclasts and osteoblasts. It's a simple mnemonic that, as you can see, this passage really, it does lay out for you what the function of osteoblasts and osteoclasts are, but it's possible that a passage won't. So that's important to know. So if we move on to the next question, from what primary embryological germ layer is bone derived? Okay, so we know that there are three embryological germ layers: endoderm, ectoderm, and mesoderm. So this is essentially a content-based question. You can't really look at the passage in this case and find anything about the germ layers. So they want you to be able to answer this question based off your own knowledge. So to answer this question, you need to know generally what types of tissues the germ layers will form. So there's essentially general trends to the type of tissue that each germ layer will form. There are exceptions, but it's good to know that the endo, meso, and ectoderm essentially are the middlemost germ layer, the insidemost germ layer, the middle germ layer, and the outer germ layer. The neural crest is not a germ layer, so we can instantly rule that one out. So if you imagine your abdomen, your ectoderm would differentiate into things like your skin and your hair and other epithelial tissue. Your endoderm, on the other hand, is going to be your middlemost layer or kind of your tube within a tube. So imagine your digestive tract or a few of your internal organs come from the endoderm. The mesoderm, which is maybe a little bit less clear, is um, our middle germ layer. And this germ layer tends to give rise to things like connective tissue, bones, and muscle. So if you try to think of yourself as three different layers: outer, middle, and inner, it's best to do that within thinking of like your own abdomen. Your outer layer is your skin or your ectoderm. Your middle layer would be like your fat, like your abdominals, your muscles, like your abdominals, like the mesoderm. And your most inner layer would be your tube within a tube, your intestines, your stomach. That would be the endoderm. So looking at this, bone would be derived from the mesoderm. And if we check here, that is correct as well. The neural crest is actually derived from the ectoderm, like it says here. The ectoderm, which we determine as our outermost germ layer, gives rise to the skin, like I said, but in addition to that, a lot of different neural tissue. And that's because of the way that the neural crest is formed. It's formed by essentially folding the ectoderm in on itself, creating a hollow tube, essentially. All right, let's move on. So this question: are the results of the epidemiology study valid? Why or why not? And what can be concluded about the incidence of ARO in the United States from the study? So we're looking at the incidence of ARO based off the results of this study. So if we go down here and we reread the part about the study, it attaches a survey to an annual political election ballot routinely over 10 years with a yearly sample size of nearly 10 million. So if we go through our answers here, we have 2 yeses and 2 nos. Yes, the results are valid. The sample size is large, randomized, and likely to represent the study population as a whole. Yes, the results are valid. The sample size is sufficient. The study is blinded, and the extensive time frame of the study minimizes the effect of bias. No, the results are not valid. Although the sample size is large, it is affected by non-response bias. Or no, the results are not valid. Although the sample size is large, it is affected by considerable bias. So I think the easiest thing to take away from this right away is that our sample size of 10 million is more than sufficient. It is large. And so I'd be hesitant right away to even check B, even just not looking into the question at all, even if you're confused about whether or not the results of this would be valid. So I would be pretty cautious to start with B, but I think really the next thing that we need to figure out is whether or not the results are valid. So in order to figure out if the results are valid here, one thing we do want to look at is sample size, which we've determined is sufficient, but also the sampling of the survey. So we know that the sampling happens on people who vote. So essentially, it's not as random as it may seem, as a randomized controlled trial would be. In addition, if we look up here, ARO, which is what they're asking about, is a congenital condition, meaning it's present from birth, affecting individuals from birth due to a number of genetic anomalies, typically resulting in death by the age of 10. So that is our biggest red flag. That is our issue here. If ARO typically causes the death of these individuals by the age of 10, how can they participate in a study at the age of, at the minimum, 18 in the United States, and the voting age, I should say. So already, this doesn't really make sense on how we could say that the results are at all valid. The majority of these people are dead before these people with ARO have died before they're even given the opportunity to complete this survey. So I'd say no to part A as well, saying the results are valid. We agree the sample size is large. Randomized, it's not overly randomized because of the age requirement, and likely to represent the study population as a whole. And this last sentence here, it can be concluded that there have been no new cases of ARO each year. That's where we really realize that this is incorrect, because of these patients with ARO die before they even have a chance to complete this survey. Then it's incorrect to say that there are no new cases of ARO each year. So that leaves C and D. Both said that the results are not valid, which we agree with, and both say that the sample size is large, which we also agree with. So it comes down to another factor. So what is different between these two? The only conclusion that can be drawn is that there are fewer cases of ARO in the United States in 2000 than 1990, or that no conclusions about osteopetrosis can be made. So essentially, for the reasons that we've already gone over, you can't really draw any conclusion about the cases of ARO in the United States in 2000, in between 2000 and 1990. We already said the results are valid, so it would be difficult to say that a conclusion could be drawn at all. So the answer for this question should be D, excuse me. So here they're looking at incidence, which is new cases per unit time, typically per year. In this case, since the results are not valid, you don't really need to look at incidence in order to figure out this question. But if you think of incidence versus prevalence, which is another measure of the commonality of a disease, think of incidence as the water flowing into a tub, and prevalence as the water as a total in the tub already. So the incidence would be like the amount of water that flowed into the tub from 5 p.m. to 6 p.m., whereas the prevalence is the total amount of water that was already in the tub in addition to the water that was added. And again, you don't need that to answer this question, but in the future, you may need to tell the difference between incidence and prevalence. And likely that'll just be something you need to know based on content. It won't be within the passage. Okay, question 4. So question 4 states that ARO is associated with mutations in RANKL and OPG. What structural or functional changes in RANKL and OPG would most likely be found in RAO? Okay, so RAO, if we remember and go back, is characterized by excessively thick and dense, albeit brittle bones. So we're looking at increased bone formation. We're looking at having essentially too much bone instead of osteoporosis, which is porous bone or too little bone. So to get increased bone formation, we can either have an increase in osteoblast activity or decrease in osteoclast activity. And in this case, we're looking at mutations in RANKL and OPG. So this diagram here shows the how parathyroid hormone eventually leads to the creation of mature osteoclasts and bone resorption. So parathyroid hormone will bind to these osteoblasts, and this this protein RANKL will bind to its receptor RANK along, or excuse me, not along with OPG, um, along with OPG being able to competitively inhibit RANKL, which it says right here. So essentially, if there's too much of this molecule OPG and it binds to RANKL, RANKL cannot bind to its receptor rank and produce mature osteoclasts. So if we're going to look here, what structural or functional changes in RANKL and OPG would most likely be found in RAO? So A says increased affinity of RANKL for its receptor and decreased production of functional OPG would each be found in RAO. So if you have more affinity between these two and decrease of OPG, its competitive inhibitor, that will lead to more mature osteoclasts, which leads to more bone resorption and more porous bones. So that doesn't seem correct. B is saying the opposite. Decreased affinity of RANKL for its receptor and increased production of functional OPG would each be found in RAO. So if we have decreased affinity between RANKL and its receptor, an increase of its competitor, that will lead to less mature osteoclasts, leading to less bone resorption. So that's certainly possible. C says increased affinity for RANKL for its receptor and increased production of functional OPG would each be found in ARO. So since RANKL and OPG as molecules essentially compete to have this result, to have the outcome of mature osteoclasts, if you have the increase of one and the increase of the other, you're essentially going to have it's going to be a moot point. Nothing's really going to result from that. So there's going to be no change in the production of mature osteoclasts. And then D is essentially the same but opposite. And then there's decreased affinity and decreased production of functional OPG. So decreased affinity for RANKL for rank will decrease mature osteoclasts, but a decreased production of OPG will also decrease its competition and in turn increase the production of mature osteoclasts. So neither C nor D can we really say anything. So the answer must be B, and it is. All right, so we'll move on to this last one. Okay, so what would be a likely effect of increased parathyroid hormone secretion? So again, if we look at our diagram here, parathyroid hormone essentially leads to an increase in mature osteoclast activity, which leads to bone resorption. So how does bone resorption affect the body as a whole? Bones are made of hydroxyapatite crystals, which are mostly formed of calcium and phosphorus. So if we look at the options here, we have increased free calcium, increased osteoclast inhibition, increased OPG expression, and increased serum phosphate. If we look at B first, osteoclast inhibition would not happen from increased parathyroid hormone secretion. Actually, the opposite would happen. So we know it's not B. For part C, increased OPG expression, OPG expression doesn't really seem to have any bearing on parathyroid hormone secretion. So this is kind of a red herring. It doesn't say anything in the passage that would suggest that OPG is somehow related to parathyroid hormone. As you can see, it's kind of down the line of the signaling cascade that leads to mature osteoclast production. So it's likely not C. So the difficulty here is between increased free calcium, and increased serum phosphate. There's nothing in the passage that would really indicate one or the other. So this is kind of a content-based question where you have to know the effects of parathyroid hormone. So like I said before, hydroxyapatite crystals are comprised of calcium and phosphate. So if we have increased bone resorption, we'll have an increase of calcium and phosphate within the blood. However, parathyroid hormone also has another additional effect on phosphate homeostasis in that it increases the resorption of phosphate within the kidneys. So although phosphate is increased in bone resorption, it's also excreted, it has increased excretion within the kidneys as a result of parathyroid hormone. So although it may seem like you can't really answer between these two, the answer here would be free calcium, because parathyroid hormone really doesn't have a big effect on phosphorus homeostasis. So if we give this a go, looks like we're right here. It says calcium and phosphate levels are typically inversely related under PTH control. According to the Kaplan books that I've read, it's really more that phosphate control is essentially a mute point underneath PTH secretion. So here we're correct as well. If you guys have any questions, feel free to reach out. We have an email here. Let me find it. Our email is here, info@premedpeers.org, or you can contact us via Slack. Reach out with any questions. And thank you for listening, and good luck with your studying.